Hi, hello, welcome back to my channel. This is Dr. Srivani Sesham. Today, let's learn about some genetic concepts. First, let us see the definition of genetics. The branch of biology that deals with study of how genes are passed on from one generation to the next, that is heredity and variations. Gregor John Mendel is the father of genetics. Gene, the basic unit of heredity passed from parent to offspring. DNA, deoxyribonucleic acid, is the molecule that carries the genetic information needed for the development and functioning of an organism and exists in double helical structure given by Watson and Crick. RNA, ribonucleic acid, complex compound of high molecular weight that functions in cellular protein synthesis. The difference between DNA and RNA being DNA is a double helical structure while RNA is single stranded. Nucleotides. These are the basic building blocks of nucleic acid. RNA and DNA are polymers made of long chains of nucleic acids. An allele or allelomorph, a pair of genes located at corresponding positions on a pair of homologous chromosomes. Alleles interact to determine a particular trait. Traits, specific characteristics of individual. Traits can be determined by genes. Traits can be qualitative, such as eye color, or quantitative, such as height, etc. A given trait is part of an individual's overall phenotype. Monohybrid. When the cross takes place between the parents differing in a single pair of contrasting characters results in a monohybrid individual, like tallness and dwarfness or red eye and white eye in Drosophila. Dihybrid. An individual which is hybrid or heterozygous in two pairs of alleles or allelomorphs is known as dihybrid. For instance, the cross between pea plants with yellow round seeds and green wrinkled seeds produces a dihybrid having yellow round seeds. Dominant trait. Dominant traits are those which are more likely to be inherited due to stronger allele expression compared to recessive alleles. These traits are expressed if an individual inherits even one dominant allele. Recessive trait. Recessive traits are only expressed if the organism inherits two recessive alleles for that trait. Now, homozygous, a term that describes having two identical variants of the same gene. In a homozygous genotype, either both genes are normal or both genes have the same mutation. Heterozygous, a term that describes having two different versions of the same gene. In a heterozygous genotype, each gene may have a different mutation or one of the genes may be mutated and the other one is normal. Genotype. The term genotype refers to the genetic makeup of an organism. In other words, it describes an organism's complete set of genes. The term can be used to refer to the alleles or variant forms of a gene. They are carried by an organism. Phenotype, the set of observable characteristics of an individual resulting from the interaction of its genotype with the environment, like the tallness or dwarfness. Now let us see each of the terms when depicted in an example. Here we can see the parental generation being depicted by the capital letters and the small letters. So the capital letters denote the dominant characters and the small letters denote the recessive characters, as well as and the parental generation is carrying the similar genes. They are in the homozygous condition. So the character that is being depicted by the parental generation, dominant is the tallness and the recessive is the dwarfness. That is why homozygous tall and homozygous dwarf are indicated here. Then when gametes are formed, the gametes are for capital T from the homozygous tall parent and small t from the homozygous dwarf parent. When a cross is made in the F1 generation, we'll get an heterozygote, that is capital T and small t, which are present as the genes in the offspring, form the genotype. And externally, what we are able to identify the height of the plant is tallness. That is why the phenotype is tall, and since the genotype is carrying two different genes, that is capital T and small t, it is heterozygous tall. So like that we have capital T if it is present, the dominant, homozygous dominant. Similarly small t representing the contrasting character for tallness, that is the dwarfness. And then in the F1 generation, when a cross between two homozygous individuals is made, has resulted in a offspring which is heterozygous. So when it is carrying two different genes like this, then this condition is called as heterozygous condition. And since the phenotype is tall, we call it heterozygous dominant. And then coming to what is F1 or the first filial generation. The first generation of a given cross is known as F1 generation. Then what is F2 or second filial generation? The second generation which is resulted by interbreeding or selfing of F1 offspring. It is known as second filial generation. Let us see some of the ratios which are formed after the crosses are made, whether F1 or F2. So the phenotypic ratio of monohybrid cross is 3:1 and genotypic ratio of monohybrid cross is 1:2:1. Now phenotypic ratio of a dihybrid cross is 9:3:3:1, while the genotypic ratio of dihybrid cross is 1:2:4:1:2:1:2:1. Then coming to the epistatic ratios, dominant epistasis shows 12:3:1 and recessive epistasis shows 9:3:4. Now coming to the next term, back cross. Crosses between F1 offsprings with either of the two parents are known as back cross. Whenever F1 hybrids are crossed with dominant parents, all the F2 offsprings develop dominant characters. On the other hand, when F1 hybrids are crossed with recessive parents, individuals with both the phenotypes appear in equal proportions. Test cross. The test cross is a cross between heterozygous F1 hybrid and double recessive homozygous parent. The test cross is used to determine whether the individuals exhibiting dominant character are homozygous or heterozygous. Incomplete dominance. A system in which the interaction of alleles in a hybrid results in a phenotype which is intermediate between the phenotype of homozygous dominant and homozygous recessive individuals. Codominance. The situation in which traits determined by both alleles of a hybrid are fully expressed. There is no masking effect or dilution of phenotype compared to the homozygotes of each allele. Multiple alleles. The existence in a population of number of alleles determining variations of a particular trait such as eye color or ABO blood groups. However, only two of these are present in any one individual. Genetic code, the sequence of three nucleotides on mRNA that code for specific amino acids. These are 64 triplets in the code. Codon, a sequence of three nucleotides on the mRNA which codes for particular amino acid. Anticodon, a sequence of three nucleotides on one of the loops of tRNA which is complementary to a codon on mRNA. Linkage, the tendency of DNA sequences that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. Crossing over, the process by which chromatids of homologs exchange portions of genetic material, usually during tetrad formation of meiosis. Chiasma, configuration made by chromatids of homologs overlapping one another during tetrad formation of meiosis, when genetic crossing over is believed to occur. Supplementary genes. These are two independent pairs of dominant genes which interact in such a way that one dominant gene will produce its effect whether the other is present or not. The second dominant, when added, changes the expression of the first one only in the presence of the first one. Duplicate genes, when the two pairs of genes determine the same or nearly same phenotype so that either of the two genes is able to produce the character in question. These are said to be duplicate factors. Collaborator genes. In collaboration, the two gene pairs which are present of two separate loci but influence the same trait interact to produce some totally new trait or phenotype that neither of the genes by itself could produce. Lethal genes. Certain genes are known to control the manifestation of some phenotypic trait as well as affect the viability of the organism. Some other genes have no effect on the appearance on the organism but affect the viability alone. These genes are known as lethals or semi-lethals. Epistasis, the interaction between non-allelic genes in which one gene mass inhibits or suppresses the expression of other gene. The gene that suppresses the other gene is known as inhibiting or epistatic factor, and the one which is prevented from exhibiting itself is the hypostatic factor. Pleiotropy, the occurrence of a syndrome of diverse effects resulting from the mutation of a single gene, example sickle cell anemia syndrome. Operon. Operon is a group of closely linked structural genes and the associated control gene which regulate the genetically controlled metabolic activity. Structural gene. The structural genes are those segments of DNA which carry codes for the synthesis of proteins. These genes determine the primary structure of polypeptide chain by controlling the sequence of amino acids during the synthesis. Control genes. These control the activity of structural genes either by induction or suppression. Regulator gene. The regulator gene produces some specific enzymes which act as a repressor substance which strongly binds to the operator gene and suppresses its activity. Promoter gene is the DNA segment at which RNA polymer binds. It initiates the transcription of the structure gene. It controls the rate of mRNA synthesis. Operator gene. It is a segment of DNA which exercises a control over transcription. The repressor substance produced by the regulator gene binds with the operator gene. Cistron, the smallest unit of DNA that codes for one polypeptide chain. Muton, the small genetic unit that can mutate is a muton, and Recon is the smallest genetic unit that can undergo crossing over. Protein synthesis, the process of synthesis of polypeptide chains by transcription of the code on mRNA from DNA template in the nucleus to translation of the code at the site of ribosomes. Transcription, the synthesis of new mRNA molecule using DNA as a template. Translation, the process by which the message of DNA encoded in mRNA is converted into specific polypeptide at the site of ribosomes. Thank you for watching, and please like, share and subscribe and hit the bell icon so that you get the notification.